Suppose 5 similarities have been found among the structures, which suppose contains benzene rings. So we'll select the 5 structures and eliminate the other 5 structures which doesn't contain. Just as simple as like that. And other way is using docking program to identify structures predicted to bind strongly. Other ways is just like docking. So we have a protein containing active sites or not containing active sites, maybe at the surface, anything like that. We have a ligand, unknown molecule. We directly bind or we directly dock to the active sites and get some docking score, and then score the molecules and screen the molecules from that. We make a threshold of the molecules that suppose if the docking score is below 100, so we delete those molecules which are below 100. We will select those molecules above the threshold. So this is one procedure of virtual screening. Another is filters remove structures not wanted in succession of the screening methods. Means there are various filters just in the virtual screening. It's just like the classifier, like remove useless. We don't just if the molecular descriptors, if the tools doesn't find out any molecular descriptors, we can eliminate those descriptors or eliminate those molecules as it doesn't contain any values in them. So we can also screen just like that. Next, main classes of virtual screening. So depending on the amount of structural and bioactivity data, which actually all we have, all the bioactivity data we all have. Suppose there is one active molecule I have. What can we do from that? Means from the database we have got only one active molecule. So from that one active molecule, how screening could be done? So from that one molecule, we can perform similarity search in the other databases to collect the other molecules. That's the term ligand-based virtual screening. So other thing is that if several active molecules are present, we try to identify common 3D pharmacophore, then do a 3D database search. A 3D pharmacophore is just a concept of a 3D molecule containing the hydrogen bond acceptors, so which will be interacting with the particular target. It's just a concept, pharmacophore. Yeah, it's not available right now. I will. Okay. So the next thing is that another thing, a reasonable number of active and inactive structures known. So we have previously 2 points was that the active and both the concept of active molecules are there. So now comes active and inactive. So we are dealing with active and inactive molecules. So what we do, we will use a machine learning technique to generate the molecular descriptors. To use a machine learning technique, and with the help of molecular descriptors, we will classify the inactive and the active data sets. So other thing is that 3D structure of the protein known, then use protein ligand docking. We can also do that. But we will be considering we have the data set which contains active and inactive structures. So we will be dealing with this. Suppose what are actually molecules contains some properties. So these are the properties: chemical properties, biological properties, intrinsic properties, pKa, log P, solubility, stability. Biological properties contains activity, toxicity, pharmacokinetics, biotransformation. These are the properties which defines a molecule, or molecular descriptors you can say. So molecular descriptors used for machine learning. So what are molecular descriptors? They are numerical values that characterize properties of molecules, as I said earlier in that previous slide. So it is being classified into 4 categories: topological, geometrical, electronic, hybrid, or 3D descriptors. So for classification in the Chemiformatics project, we used the pharmacophore fingerprints, weighted burden number, and the properties. With the help of some tool which will be given by Vinita, the pharmacophore finger contains 147 descriptors, weighted burden number about 24 descriptors, properties of 8 properties contains just like X log P, is log partition coefficient values, molecular weight, and almost 8 descriptors are there. Now we have the descriptors right now. So what to do with the descriptors? So we will do mining the data. So why mining? So we have the data, means the molecular, the descriptors values. From that, we get the information, and from that information, we get the knowledge. That's what is that I have represented there. The main aim of a user is always extract knowledge. What is data mining? It's extracting the knowledge from the data to understand the data perfectly. So now, data mining is one of the key steps in knowledge discovery process. Although sometimes confused with knowledge discovery itself. It's always a process because data are generated continuously. So this continuous is a process. It's not the term knowledge discovery. It's process. Okay, data mining in cheminformatics. Data mining approaches are integral part of cheminformatics and pharmaceutical research because we will be dealing with totally artificial intelligence right here in the cheminformatics team. We will be building models and imply it. We will be building mathematical models with this data mining. This will it will increase due to the increase of computational methods for biology and chemistry. So many data are available, biological data and computation and chemical data. We will use this data in data mining. So data mining has also found major use in virtual screening, which I will be telling which I have told you. So this is the basics, means the paradigms of the data mining. So here it is maintained that some classifiers names, neural networks, Bayesian networks, decision trees, support vector machines, and instant-based regression classification. So these are actually the classifiers, the algorithms behind the artificial intelligence. So we will be dealing with these algorithms to separate the active and the inactive data. Right? So in our project we use these type of classifiers, naive-based, J48, random forest, and functions is SMO. So these are the 4 classifiers which are being tested worldwide, and they are being selected one of the best classifiers. And it's because that these classifiers have been tested, and they are selected the best classifiers. Yeah, there is a lot of classifiers in the tool which will be used, which will be said by Vinita. The Veka tool will be introduced to you by Vinita. Almost more than maybe 50, I think. Around 40 classifiers are there. So now we have did some little bit other classifiers to test our data, but the results were not promising so much.
So other class of drugs, apart from TB, other class of drugs can be analyzed with these parameters? Yes. There is fully validated? Yeah. Okay. Then okay.
So now the workflow model of the cheminformatics will be described by Anurag Pasi from IGIB. Thank you.